Hi, my name is Allison Kaplan. I'm Director of Education at the National First Ladies Library, located at the National First Ladies Historic Site. For today's Ladies in the Laboratory, we're going to be looking at two women scientists, Barbara McClintock and Rosalind Franklin, who studied the basic human building blocks of life, genes, and DNA. We're going to start with Barbara McClintock. Barbara McClintock studied genes, and what genes are is the basic unit of heredity, a special type of DNA that codes something. Genes determine what traits are passed down from a parent to kid, like hair color or eye color. A single strand of DNA can contain thousands of genes. Barbara studied genes in corn, but today we're going to look at strawberries and attempt to extract some DNA from strawberries. You'll need 1-inch layer of isopropyl alcohol, 1 teaspoon of salt, 1/3 cup of water, 1 tablespoon of dish soap, a clear glass cup or beaker, a funnel, and a coffee filter, strawberries with the greens cut off, 1 plastic bag. The first step in this project is to take a plastic bag that has a nice ziplock to it. Take your strawberry, cut off the greens at the top, seal your bag nice and shut, making sure you get all the air out of the bag. And then you're gonna squish down your strawberry. So you're gonna try to break it down as much as you possibly can. So we're breaking it down into those little bits so we can get down to that DNA, down to those little cells that build up the strawberry. So you don't want any chunks, you want it to be really kind of gooey and goopy. And you'll do that with your hands and break it down as much as you possibly can. Now that I have my strawberries all squished down into goo, I am going to add 1/3 cup of water, a teaspoon of salt, and 2 tablespoons of dish soap to create a mixture. I'm gonna make sure my bag is sealed up nice and tight. And then I'm gonna continue, and this time carefully, because you don't want the goop to get all over the place, all over your kitchen or house. And you're gonna squish more and make sure it's really mixed up. And this solution is gonna help that those cells separate in my strawberry and break it down and break it apart even more. Now that we have our strawberry solution, the next step is to pour it into the beaker to make sure that any little chunks that might be left are removed. Now I'm pouring it through the funnel, and the coffee filter is picking up any of those extra chunks of strawberry that didn't break down completely. And I'll give this a little time to pour through. So you can see that the liquid is going through the filter, and anything that's not a big chunk of strawberry is coming down through my funnel. So the next step is to take that inch of isopropyl alcohol and to pour it in. And as you pour it in, you're gonna let it sit for a little while, and you're gonna start to see it separate. And there's gonna be a little cloud of white wispy material that forms at the top, and that white wispy material is going to be your strawberry DNA. So let's pour this in and see what happens. And we'll use this little pipette to kind of move it around, and you can start to see, you can pick it up a little bit, and you'll see that it picks right up out of the water. And this long strand here is actually the strawberry DNA. So you separated the DNA from your strawberry. How cool is that? Rosalind Franklin is the scientist we're going to look at next. She was the first scientist to take a photograph of DNA, and she did that with an X-ray machine. We are going to build our own DNA strand in honor of Rosalind Franklin. DNA is the essential molecule for life. It acts like a recipe holding instructions that tell our bodies how to develop and function. Rosalind Franklin took some of the first photos of DNA. A strand of DNA is much, much thinner than human hair. You can't even see it with a microscope that makes things look almost 1,000,000 times bigger. She worked in an X-ray lab to capture the image called Photo 51. With her picture, scientists made a Tinker toy-like model to show that DNA is shaped like a twisted ladder. This shape is called a double helix. So the first thing you're going to do is cut 4 long strips of tape. You can use electrical tape or duct tape. And so you're going to put it sticky side up, and then you are going to gather straws. Now you're going to need 4 different colors of straws, or the other thing that you can do is take a Sharpie marker and color them. You're going to need tape to tape your straw pieces together, scissors to cut the different colors apart, and you're going to need your pencils so you can twist your DNA strands in place. DNA is made of chemicals called bases. The steps of the DNA ladder are pairs of bases. Here they are: adenine and thymine, cytosine, and guanine. You're going to take your straws and choose which colors represent each of these for you. So for adenine, I chose red. Now, adenine and thymine can pair, but they can't pair with these two. So I can't put thymine and cytosine together. So I'll take my red straw and my blue straw and cut them into equal parts. And then I will tape them together, like so. Once you're done creating your pairs, you'll have about 5 of these, red and blue, or whatever color you choose, and 5 of the green and purple. For the next step, we'll take our long strands of electrical tape, and you can make them as long as you want. So you could even do a super long DNA strand. And I'm going to take a pencil and set it down on either side. And then I'm going to start to lay my strands, and I put them about equidistant apart. And even though my red and blue have to pair together and my green and purple have to pair together, there isn't any set pattern. The DNA can have many different patterns. And I will keep laying these down until I get to the end of my ladder here. Let's add one more. And for the final step, I will take my strands of tape, and it's okay if they're not perfect. Scientists are always testing and experimenting, and sometimes you'll find that something you do wrong, an error, is a new discovery. So I will take my strands of tape and lay them down on top to make a sandwich with the straws in between, like so. And then I'm going to roll my pencil up a little at the bottom and This one looks good up here. So I'm all done. I can squish down the tape pieces and I can pick up my DNA strand and twist it to form that double helix. Thanks for joining us for our final installment of Ladies in the Lab for this school year. Happy experimenting, and we will see you next fall.